about epidemiology. So, normal microbiota is the term used for normal flora. And right, normal flora is beneficial to us, and the bacteria that are considered normal flora, we're beneficial to them, right? So we consider it a mutualism or mutualistic type of symbiotic relationship. Symbiotic relationship means the two organisms are living at the same time, right, together. Normal flora is another word for normal microbiota, maybe the old term for it. If you have a parasitic type of relationship, that would be like a virus, right? Viruses never really help us when they infect us. A pathogen is a microbe whose relationship with host is parasitic, right? It involves causes us a problem. And just the fact that we have bacteria in our body that don't belong there that can be the problem, right? If you get bacteria in your, say in your bursa sac in your knee, they don't belong there, so that's going to be a problem for your knee. Pathogenicity, right, is just how big of a problem they are, right? We considered COVID-19 at the beginning very pathogenic. Today, it's not so pathogenic, at least in my opinion. Other people may differ in that opinion, but so that even pathogenicity can change, and really that's what happens with viral outbreaks like this. They tend to be fairly pathogenic at the beginning, and then over time, viruses evolve and become less pathogenic. Even Ebola has. Say 20 years ago, 30 years ago, Ebola, you had almost 100% chance of dying from Ebola if you got Ebola got infected with it. Now it's, it can be as low as 50%. It behooves the pathogen to keep you alive. If it kills you too quick, then it's not going to spread. Another way of putting it. And virulence and pathogenicity, right, refer to the same thing. So in this chapter or section, rather, right, we think of the host, then it's usually the human being. If we talk about animals that come down with some sort of bacterial infection that can, say, infect us, then we might consider the animal the host, but for the most part, right, it's the entity that has the pathogen. So mammals are warm-blooded, right? They maintain a constant body temperature. So that's helpful for bacteria, right? We've already emphasized the fact that most of the bacteria that are normal flora in the body, or that maybe could cause infection, they want to grow best at 37 degrees. That's their optimal temperature. Mammals provide plenty of nutrients, and they maintain constant pH, just kind of like they maintain a constant temperature for both of them, right? Now, in this section, there are lists. Lists are important, regardless of, keep that in mind. Why, good question, what makes mammals good? Which of the following make the reason why mammals make good hosts? Any of those three would be correct. So we expect to find bacteria on our skin, our respiratory tract, gastrointestinal tract, urethra, genitalia, even in the ear, external ear. So there's certain places we know we find bacteria, right? And there's no way of getting rid of the bacteria. You can't sterilize or any type of body part or anything like that. Human Microbiome Project, I just mentioned this just to say that we're still looking for places where bacteria might exist that we don't expect to find them. It's probably going to be unusual though. But even the sinuses, I'm not sure exactly at what point in the sinuses you would consider it sterile. You have a sinus infection, that's a different point, but you know, some of these, you might find them in the middle ear, for instance. You're probably not going to find them in the blood. So, what we want to do, the most of this section now is going to be on the stages of a bacterial infection. Right? We're really not going to talk too much about viral infection. This mainly focuses on bacteria. And the first thing that has to occur, right, is they have to gain access. So when we do section 13, which will be the next section, we'll start that next week. We think of the innate immune system that has barriers like our skin, our mucous membranes, that keep bad bacteria out. And even the fact that we have normal flora there helps keep the bad bacteria out too. So if they get past that barrier though, we call that the portal of entry. And I think I said before, you know, sometimes, you know, you can say that some pathogens, if they don't enter through the correct portal, they're not going to cause an infection. Once they get in, then they have to be able to stay where they are. So you call that attachment. Then the first thing that's going to happen is our immune system is going to attack. So we'll talk about the ways organisms or pathogens sort of get around our immune system, evade it, avoid it. Then we'll get into the mechanisms of causing damage or the problems associated with having bacteria in our body that shouldn't be there. And then if they want to go on to another person, then they exit and find an entry point in somebody else. So portals of entry would include things like your skin, getting through the skin, right, if it was a cut or something like that. Even the eyes, nose, mouth, GI tract. And like I said, you know, a pathogen, for the most part, right, if they don't get in the way they normally do, then they probably aren't going to cause the problem. Right, so you might say like it's sexually transmitted, right? They get in through genitalia. You have bodily fluids are mixed. If that doesn't occur, you're probably not going to get HIV. Or if you use a needle, right, that's getting through the skin, that's a portal of entry, say using a contaminated needle. Now, if you do get some bacteria, or in this case, we can even include virus, you might need to have so many get through the portal at one time. I think they did a study with gonorrhea, and it was about 1,000, took about 1,000 bacteria. And you can even describe it as an ID50, which would mean if you had an ID50 of 20, then it wouldn't apply to this gonorrhea, but it means though that if you get, say, 20 virus in your body, then you have a 50% chance of coming down with an infection with it. So there are times when bacteria get into our bloodstream and we don't know it. I mean, if you just cutting your skin, there are bacteria going to get into your bloodstream, but your body gets rid of them. It doesn't establish an infection. Same thing with COVID-19 or even the flu. Most people that get HIV don't even know their acute infection. They might have HIV, an active infection with HIV for like a week. That's what they used to teach us, at least. They get over it. They might have felt bad for a couple days, but there's not a lot of symptoms. And that can be like that with other viruses, right? So like when children, they would call it, right, they're asymptomatic, right? They've got COVID, but they aren't sick. We really don't even know how many virus are in their body, but they have a positive COVID test. their body is able to fight off the COVID-19, so they don't really, and that's the whole purpose of immunizing, really. It's not to protect you, if you get the flu shot, it's not so much to protect you from getting the flu, as much as it is to keep you from dying from the flu when you get to be 80 years old, right? It's not, you know, immunity, we're not really getting a good definition of what immunity means through all this stuff that we're learning now. I'll give you a pretty good definition of it when we get into sections 13 and 14. But, you know, why people can't say children are immune, I have no idea, because that's an accurate statement. So, you can use a capsule, bacteria make a capsule, right, which is a secretion. And it helps the bacteria stick together and stick to surfaces like epithelial cells. And the fimbriae is also used. You would think of the bacteria as having a ligand on the surface. Right, let's just say it's a triangle. I'm making it much larger than it would probably be. And then, right, that would be able to say, this is a cell, an epithelial cell, and the ligand sticks to a receptor on that cell to somehow attach to it. Right, so we think of the microbe as having adhesions or ligands, and cells in our body act as sort of like receptors for those ligands, and that's how. Now, see, the ligand could be on the fimbriae, whereas the capsule is more like glue, right? It's sticky, gooey stuff. So it doesn't really meet that kind of scenario. It's not ligand to receptor. It's more like a glue. Now, a couple things need to occur, right, is to get the bacteria to divide well, is they need to overcome, or it's like something like evade phagocytosis. So let's turn this now into a host cell. Let's say it's a white blood cell for the host. And in section 13, we'll talk about phagocytosis. But white blood cells or phagocytosis, that means they eat bacteria like this. And the way they do it is they have a receptor that binds to a specific ligand on the bacteria. And we'll call them PAMPs. Now, if the bacteria secrete a capsule, let's say that's the capsule now, this circular, it can cover up this ligand and prevent the white blood cell from attaching to the bacteria. So we think of the capsule as a protection the bacteria has to prevent phagocytosis. Now, the other thing that occurs, I'm drawing the bacteria way too big, but the bacteria gets into, say, a vesicle, and then in that vesicle, the white blood cell needs to kill it and then break it down. It's real difficult for white blood cells to digest Mycobacterium tuberculosis. And Streptococcus pyogenes even has what's called an M protein, so that makes it more resistant to digestion. And what can happen is the bacteria just start dividing, right, inside the white blood cell. Instead of being eaten, they start to eat the white blood cell instead. That's not good. At least for the white blood cell. The bacteria are happy with that arrangement. So host defense mechanisms means our immune system. When a white blood cell phagocytizes it like this, right, there's going to be two main components to immunity. There's the innate immunity, which includes phagocytosis and your barriers. They call it innate because you're born with it. The other main category is the acquired immunity. Acquired immunity reacts to foreign antigens. So when you get vaccinated, they put an antigen into your body so that your body mounts an immune response to the antigen. Your innate immunity doesn't react to a foreign antigen or pathogen. It just knows that this bacteria is bad and eats it. Now what's strange is like they don't eat our normal flora. They can tell the difference between good bacteria and bad bacteria. Because normal flora, there's lots of normal flora in our body. And if we don't have it there, it generates problems. So what's an antigen? Right, if you're a virus, you might have, right, so you have spike proteins. And on the spike protein, there might be 5 or 6 antigens on there, right? Now let's say the virus mutates. Mutations cause the antigens. Say instead of being a circle, let's say now we have a square shape. It's a little less subtle, more subtle than this. So if we have an antibody that we've generated to this virus that binds to the circle shape, the same antibody cannot bind to the square shape. That's what's happened, say, with COVID-19, with the Delta variant, right? There's been several variants, but we're worried, right, that the variant is changing. I want to move this a little bit. That the variant changes shape, and now even so, if we've been vaccinated and we generate antibody to the earlier versions of COVID-19, we get the Delta variant. Now this antibody that we generate to the vaccine is no longer effective. That's the result of antigenic variation. Why don't we have a vaccine for HIV? Right? Because it mutates so quickly that once you develop the vaccine, it becomes obsolete before you can actually use it, or if you start using it, it becomes obsolete. So the antibody, right, that's generated. Now there are organisms that they're known for their antigenic variation. The influenza virus, the coronaviruses do. They're called HIV is one like this. They're RNA viruses, and RNA viruses are notorious for this antigenic variation. That means they can produce variants very quickly, which makes the vaccination sort of impossible. At some point, I'm not sure when, but someone's going to say, the COVID-19 vaccine needs to be redone. I mean, why they haven't even started that, I have no idea. So I kind of already explained this concept of a capsule, right, blocking phagocytosis. So now we want to get into, once the bacteria, say, get into our body, then, right, what sort of problems can they have, right? Once they establish the infection, right, they can secrete enzymes, and those enzymes can digest parts of our body, right? So if you have a flesh-eating bacteria, the bacteria isn't really, they don't have teeth, they're not chewing away, they're releasing enzymes that break down your tissue, and they can happen very quickly. Luckily, it doesn't happen a lot. So exoenzyme is an enzyme that's secreted. Coagulase causes clots. Bacteria kinase is kind of like the opposite of coagulase. It's an enzyme that dissolves clots. It's even used for stroke patients. Streptokinase. Hyaluronidase is an enzyme that breaks down hyaluronic acid. It's found in synovial fluid and other places. Collagen is a protein in most of our tissue that helps kind of hold the cells together. So this enzyme breaks that protein down. Okay, so if bacteria that can release these types of enzymes cause big problems. Not all bacteria do. If you get an E. coli infection, right, you're not going to have too many problems with these kinds of exoenzymes. Now, the exotoxin is a toxin that's secreted by bacteria, right? An enzyme digests things, breaks things up. Toxins are just toxic. They get into a cell, they don't necessarily kill the cell with the examples I'm giving you, but they cause the cell to do things it's not supposed to do. Right, we'll get into a couple examples. Now there's also an endotoxin, and I talked about this back in section 4, right? It's only found in gram-negative bacteria. It's a lipid A component. So exotoxins are secreted. An endotoxin is like inside the bacteria's wall. So when the bacteria die, it's released. Luckily, it's not too toxic. So an exotoxin is a protein. There are many of them. They're protein. They are what they're referred to being heat labile, so you can use them in when you make. I'm going to give you some examples of making vaccines with toxins. So if you do that, you destroy their ability to be toxic. They're very potent. Botox is an example. It can be a big deal if you get too much of it injected into you at once. They have specific tissue, right? They bind to a cell that has a specific receptor, and only those cells that have that receptor are affected by the toxin. So if they're a neurotoxin, then they affect neurons. If they're an enterotoxin, they infect the cells in your gut. So there's 3 categories. Category 1 are probably the most notorious. They have 2 parts. They have an A and B portion. You don't worry too much about the details of the A and B. You know, the B is called binding. That's what causes it to stick to a specific cell. Then the A portion is what causes the effect. So like for instance, right, with botulinum toxin, once it gets into the neuron, then it prevents the neuron from firing a signal to a muscle to contract. The tetanus is a neurotoxin, right? It affects neurons too, but it has the opposite effect. It tells the cell to keep firing and causing tetany. Tetany is basically your muscles, all your muscles contracting at once. Sounds horrid. I mean, I get a cramp in one muscle and I freak out. And now that I'm getting older and I get these little weird cramps in my foot, like my toes do this, like how do you get rid of a cramp on your foot, like that? Most of the time you can stretch it, right? But how do you stretch your foot? I'm learning. It hurts. And cholera toxin, right? It only affects the epithelial cells in your intestine, and it causes them to release water instead of absorbing water, which is basically what they're there to do. Keep you from having too much water in your stool. So category 2 exotoxins are membrane disrupting. That just means they lyse cells. They kill them. If you're considered a leukocyte, then you're killing white blood cells. Whereas if you're a hemolysin, you're killing red blood cells. Now, in the BAP plate, they use red blood cells. They're actually that plate is there to detect hemolysins that cause alpha hemolysin or beta hemolysis of red blood cells. But that's what's causing that on the blood agar plate. And then category 3 are referred to as the superantigens, and they give an intense immune response. Like toxic shock syndrome is an example of that. You know, it's not so much that the bacterial infection is causing the problem. They're releasing this toxin, and it's causing your white blood cells to react as if you were getting, you know, attacked by the worst virus in history. It's not the same as an allergic reaction, but that's kind of what happens with allergic reactions too. When you get this, it's called the cytokine storm, and people with COVID-19 can end up with that too, and that would be probably very similar to this. It's the intense immune response that kills you. There's so many cytokines being released by your white blood cells, it just doesn't know what to do. Your body just starts trying to do too many things at once. So toxoid is what's used in the tetanus and diphtheria vaccines. So when you're given these vaccines, they're injecting you with an inactive toxin. Your body produces an antibody, right, to the toxin, and that's what you're not trying to prevent or kill the bacteria with the antibody. So let's say the toxin has a square shape, and then when you inactivate it, let's say it gets to be a circle shape. And you develop an antitoxin antibody. Now it changes shape, but this antibody can still recognize the active toxin. So if you get infected with a bacteria that releases that toxin, then the antibody neutralizes the toxin so it doesn't have its effect. Now you're supposed to get a tetanus vaccine every 7 years, right? I mean, that's what they've told me and everybody for I don't know how long. And I'm beginning to wonder if that's really necessary. But you don't want to get tetanus, for God's sakes. So, but that's what happens, right? These vaccines do not prevent you from it's not that you're mounting an immune system response to bacteria, it's to a toxin that the bacteria makes. And it's the toxin that causes the problems, or could kill you, right? It's not really the bacteria doing them. It's the bacteria that's releasing the toxin, but the toxin is causing the damage. So the endotoxin is not secreted. It's only found in gram-negative bacteria. It's found in their cell wall. All right, we talked about the LPS being a component of the cell wall. Lipid A is the specific. So that's the name of the endotoxin, lipid A. So it's released if you're giving somebody antibiotics, the bacteria begin to die, then your blood pressure may come down some. You might develop, your fever might go up a little bit as a result of the endotoxin. It's not going to kill you. So we went through the course of the infection, and then these are what you refer to as portals of exit. Your eyes, nose, mouth, blood, feces, urine, semen would all be considered, an exit way of exiting your body. There are probably others as well. Right, so we went through the course of an infection, starting with the entry and then the exiting. We said that the bacteria had to attach in some way. then it would have to survive an immune response. So, phagocytosis is one of them. Antigenic variation was another type of avoiding it. You could still mount an infection. It's, you're still going to get infected with pathogens in your bodies. They might not be able to avoid the immune system all that well, and it still causes an infection. But they're all working together. So at some point you hope, you get over it, and then maybe before you give it to somebody else. So that's the disease portion of this section, and then the next part is the epidemiology terms you should be familiar with. So a systemic infection is one that's found in a couple parts of your body. It might be your blood, it might be your lymphatic system. It would spread from a localized infection, right? So the local is where it may have gained entry into your body, and you're hoping that it doesn't get to the systemic state. Most infections probably don't. The problem with a COVID-19 patient that really has issues is it doesn't stay in their upper respiratory tract, right? It moves in the body. And that's when you probably really have a lot of problems with COVID-19. So there's a primary infection, right? That's one organism, and it can lead to the secondary infection, which means another organism establishes an infection. So it could be, their primary could be bacteria that leads to a viral infection, or it could be a viral infection that leads to a bacterial infection. Or in this case here, it could be, right, this bacteria starts off as a urinary tract infection, and then it leads to a secondary infection in the vagina. So, as long as it's two different organisms, then, one's a primary, one's the secondary. A good example, the best one probably, is you get the flu, and then you die of a secondary infection. And that could be happening with a lot of these COVID-19 patients, right? They have a COVID-19, they get a secondary infection, they never even find out they do, they just die. And, they probably aren't really looking too hard into the cause. It's the same thing with the flu, right? If you die from, you get the flu, and I'm sure a lot of the times during flu season, people die from the flu, they're not really even, do they ever really get a flu test? They may die, and they're not even counted as a flu death. That's not the case with COVID-19, I'm sure of that. So this is another example of some lists, factors that affect the influence of the infectious spread of the infectious agent. I'll explain what reservoirs are. We already really talked about portals of entry and exit, and then I'll get into different modes of transmission. So the definition of a reservoir is a source of the infectious agent. That's where you find it. It could be the fruit bat for Ebola in Western Africa. It could be the hantavirus is found in rodents in the Southwest. So there's a reservoir. Those are sometimes referred to as vectors. Human reservoirs are the carriers, right? They're spreading it. They can be subclinical, you don't even know someone has COVID-19, for instance, and they're carrying it. Then there's animal reservoirs. You consider something like rabies a zoonotic disease because the rabies virus infects animals, but that same virus can infect human beings. COVID-19 can infect, or the coronavirus can infect bats. So it's a zoonotic type of disease. Right. So anthrax is another example. You know, ticks don't come down with Lyme disease, but deer do, or they carry the bacteria, and then the tick transfers the bacteria to us. If you got rid of the ticks, you probably would never get Lyme disease. If you got rid of all the deer that carry it, the bacteria, you could get rid of Lyme disease that way too. Then you can find sometimes soil and water are contaminated with bacteria that they can act as reservoirs. Three main categories. Then there are modes of transmission. There are three main categories for that: vehicle, vector, and contact transmission. I'm going to talk a little bit about vehicle and vector in a minute. I just wanted to group all three of them together. Then there's three types of contact transmission. There's direct contact or indirect. Direct would mean you get it from another person, or even a baby getting it from a parent. And indirect would be through a fomite. So that could be the surface of the desk in here. It could be your phone. Inanimate object passed from one person to another through the object, right? Wash towel. You consider these objects fomites, right, in epidemiology terms. I should probably have a fomite quiz question or test question about that. Pretty simple concept. Then the droplet, now this has gotten a lot of attention with COVID-19. Technically, I think you would see in most textbooks less than a meter. If you got a droplet, a sneeze from one person to another, then it would be considered contact transmission. I suppose you could probably stretch that to 2 meters or 3 meters in the current situation. Right, 6 feet now I think is the distance, or 3 feet. It changes. So that's just a, you know, arbitrary measurement. Then there's 3 different vehicles. You can say if something is spread in the water, then it's waterborne. We wouldn't consider COVID-19 an airborne problem, right? You need to be, it needs to be spread. If it could survive on the surface of these countertops, that would be different. But I think if you guys taken the homework assignment about COVID-19, there's a question about why wouldn't you expect COVID-19 to be transmitted on the surfaces? If you kept the surface moist with some water or something, yeah, that could potentially, the COVID-19 could survive. But any virus, right, see this is a virus without an envelope. But if we got rid of the, right, if we surrounded it with a membrane, right, then the membrane needs to remain in water, right? There needs to be water on both sides of the membrane. So if you dry this up, then you break down the membrane and the virus is no longer contagious. It's neutralized. So when they were saying back, you know, a year and a half ago that COVID-19 could survive for days on the surface, I knew that was just had to be crap, right? This couldn't happen. Based on other viruses that have envelopes, you could make that assumption. So foodborne, right, you get it through, it's usually food poisoning. It's found in the food. Then there's 2 types of vectors. there are vectors that you consider mechanical. And a good example of that would be like if you know, say, if you had a dirty needle and you got injected, you know, and it punctured your skin, then the virus from that dirty needle could mechanically give you the virus, right? If this fly landed on some poop and then landed on food, it could mechanically transfer the bacteria to the food. If it's a biological vector, then the mosquito doesn't come down with the disease per se. They're, you know, it says the vectors are infected. It really means that the pathogen is able to multiply inside the mosquito. So it doesn't get, the mosquito that causes malaria, they don't get malaria like we do because they're a different organism. But the parasite that causes malaria can grow in number. So it's just not the fact, like some mosquitoes can't transmit malaria because if they bite someone with malaria, they don't multiply. If that mosquito bites someone else, it doesn't transmit the malaria. You don't get enough of them transferred, you know, in a mechanical way that way. So another way of looking at it is, it has to be the right mosquito or the right fly. Right? It's, like for this Anopheles aegyptia, right, is the only mosquito that can transmit dengue. Another species of that same mosquito wouldn't, because it wouldn't be able to multiply. And I don't expect you to know the list here, but this is a list of, you know, say, biological vectors that can transmit these types of diseases, right? They have vectors. Most of the time it's mosquito or some type of fly. Ticks are another good one too, because they bite, you know, they puncture your skin. So there's a few terms here: an endemic, epidemic, and pandemic. So this example in the blue, right, shows an endemic occurrence, and I think this is the hantavirus that infects mice, and you find the mice only in the Southwest. So because of the mice, that acts as the reservoir, then you would say the hantavirus is endemic in this location of the United States. Now, rabies is endemic to all of the United States. There's always a small amount of those virus that exist. Now, if we got rid of rabies and if there was no longer a reservoir of it, then we would no longer claim the United States is endemic to it, but it is. If we got rid of this reservoir here that causes, you know, we got rid of the mouse that harbors the hantavirus, then we could get rid of, you know, hantavirus. Now, for this to become an epidemic means it's growing in number. Right there, it's getting bigger. You know, it might stop getting bigger at some point. So an endemic means it's always there. An epidemic means there's an outbreak. A pandemic means it's an outbreak that's worldwide. A communicable disease means that you can pass it from person to person. That's just pretty straightforward. All right. A non-communicable disease is not transmitted from person to person. So like food poisoning is a good example of that. Any kind of disease that's, you know, say congenital, you're born with it. You know, of course, you don't pass something like that on person to person. And then rabies is a good example of endemic, pandemic, worldwide. just already explained that. Now there's a common source epidemic, and that would be something that's very quick. And it doesn't have to be that big. You know, it could be just a picnic or a restaurant, right? They don't cook the food properly. They might not put it in the refrigerator when they're supposed to overnight, you know, things like that. But the incidents, right, you say everybody that ate it for a couple of days are sick, but they don't transmit it to other people. So the incidence goes down quickly. A propagated is when this one does get spread from person to person, and usually you see a curve like this, but you know, with corona, there are peaks and stuff like that, and a lot of times that may have to do more with there's a different variant that causes those peaks. So I'm pretty sure corona, this COVID-19 is just going to keep mutating. We've had coronavirus for a long time, and COVID-19 got a lot of attention. It's not going away, you know. No matter how much we vaccinate, the vaccines are going to become obsolete in a hurry. I have a feeling it's already obsolete. You know, everybody's, you know, they're getting reinfected, or if you've been vaccinated, you're getting infected. Once you get infected with it, your acquired immunity and your innate immunity both will probably protect you even for variants, so that it's not lethal, any of the variants that pop up in the future. I think what made this one so lethal was the fact that it was made in a lab. You know, it didn't occur naturally like most of the other ones do. So the morbidity rate refers to diseases per 100,000 in a population. Mortality is the death rate per 100,000. The index case is the first case. This would have been really nice to know for COVID-19. If we had gotten on, we were able to investigate China a little better. You know, the first case of COVID-19 could have been in like September of 2019. For all we know, could have been that early. And then I talked about primary can lead to a secondary infection already. Septicemia refers to the fact that there are bacteria dividing in the bloodstream. It's a little bit different. Bacteremia just means that there are bacteria found in the blood. Toxemia refers to toxins in the blood. Or viremia, there are viruses in the blood. So these are just more pretty self-explanatory terms, I think. So if you get a question about these, it'll be verbatim, the definitions, right? There's lots of definitions, and there are lists. So there's another list, for instance. Methods to control communicable diseases: patient isolation, quarantine, kind of the same thing. Immunization, hopefully. All right, papilloma, hepatitis B are common examples. Flu vaccine. And then vector control, if it's spread via the vector, right? So mosquitoes, get rid of mosquitoes, then, you know, I hate mosquitoes, you know, I hate getting bit. They pass a lot of things. Luckily, we don't have a lot of big problems like that in the United States. But. So, nosocomial is the old term. I think I've already mentioned this before. Healthcare associated is the new term. if you get an infection at a medical facility, right, it would be referred to as healthcare associated. Nosocomial infections is the old word use, right? And because you're in a hospital and you're sick, you might even be given antibiotics and stuff. You're more likely to come down with another infection. It wouldn't necessarily mean it's a secondary infection, but just because you're in the hospital, now they check people nowadays for MRSA when you are admitted to hospitals to see if you're already a carrier of MRSA. They don't want to be sued in the future for you getting MRSA while you're there, right? They don't want you to get a healthcare associated MRSA infection. And they might do something to prevent that MRSA from getting to other patients in the hospital, right? So they actively work at, so if they do get sued, they want to be able to say, well, this is our protocol for keeping MRSA from being spread in our hospital. That would probably be real important for their defense. So this is a list for control of healthcare associated infections. All right, aseptic technique, gloves, masks, washing hands, education of staff, use of sterile instruments and gloves. It's another list. And then these are, I'm not sure what year this is from. I've had this on here since I can remember. A list of sexually transmitted diseases. I think there's a new acronym for it now. Instead of sexually transmitted, I can't remember the name. I don't expect you to remember the numbers, but in case you're curious,